There'll be 50 questions, 2 points apiece, so it's worth 100 points. Fold your papers lengthwise down the middle, okay, like a hot dog. Right. Okay, tomorrow when you answer questions, be as complete as you can with your answer. Be as specific and complete. If the pointer is on simple squamous epithelium and the question is identify tissue, and you write down simple squamous, that's wrong. Simple squamous is not a tissue. Simple squamous epithelium is a tissue. Same thing for the connective tissues. So be as specific and as complete as you can. If you have questions, you can star it and write something on the back. As far as spelling goes, close is good enough for me. Okay. Close enough that I can understand what you've written. I have always had trouble with spelling, and so I kind of fall back to the words of Andrew Jackson: it is a poor mind indeed that can only think of one way to spell a word. Okay, although one of my colleagues was grading lab practicals for a student that was taking the anatomy course that used to be taught through UNL. The laboratory is taught here, and she was correcting this lab practical. The kid had gotten them all wrong except one, and he had misspelled it. And she asked the professor, she said, Dr. Johnson, so-and-so got them all wrong except one. Should I give him, but it's misspelled, should I give him credit? And the answer from the professor was, hell no, this is a university. Okay, well, what organ do we see right here? Thick skin, or integument, thick skin. And that light line that we see right there is what? Stratum lucidum, and so that makes this stratum corneum. It won't be that simple tomorrow. Okay, and what are these things? These things sticking up right there, and right there. Right. Dermal papillae, and these sticking down are ridges. And what is this thing right there? Seb. Right. Okay, it's sebaceous gland. Okay, what's this layer that we see right there? Stratum granulosum, and that layer is stratum lucidum, and this layer right in here, stratum spinosum. And what's that thing? Meissner's corpuscle. And the layer that we see right here is what? That would be the papillary layer of the dermis, and below that would be the reticular layer. Okay, this layer right here, this is again what? Stratum spinosum. What is that thing right there? That thing, what is that structure we see right there? And there's one right there, one right there. It's a nucleus. Okay, this layer right here, stratum corneum. And this specifically would be what type of skin? Thin skin. Okay, and so the layer we have right here would be the papillary layer. Below, just the layer just below that is what? Reticular layer. Okay, well, we've seen that one. Let's go to the next one. And I actually have another carousel with a few more slides in it, so when we change this one out, don't run off. Okay, what is this structure or organ? This is actually an organ as well. That is what? Chinean corpuscle, and how about that right there? Okay, americrine or eccrine sweat gland. There's the duct right there. What kind of tissue lines that duct? Looks like a stratified cuboidal. Okay, so we have the glandular portion, the duct right there, and this tissue. What is this tissue that we see right in here? Loose connective tissue. And how about this out here? Dense irregular connective tissue. Let's go on to the next slide. Well, let's go back a slide here. These eosinophilic areas you see right there belong to what kind of cell?
Myoepithelial cell.
Myoepithelial cell. Okay, and what gland do we have right here? That's an apocrine gland. Okay, and again we have what? Sebaceous gland, and we have a little slip of a tissue right here. It's the erector pili, which is made up of what kind of tissue? Smooth muscle. Okay, and this is not really quite fair, but we have kind of a survey view, and we see this layer right there, and that entire layer is what? Endocardium. Good. I'll have to take that one off the lab card. Okay, what organ do we see here? Elastic artery. And that layer right there is what? Tunica intima, and that makes this the tunica media. Then out here we have tunica adventitia. And how about this structure we see right there? Collectively, that would be called what? Vasovasorum. And how about these little squiggly lines we see right there? What are those? Elastic fibers. Hence the name elastic artery. And that layer again is what? Tunica intima. Okay, and another elastic artery. And this time, what is this layer right there? Tunica intima. Just turned around. There's the tunica adventitia. There is the vasovasorum. Okay, and this line that we see right there, what is that? Internal elastic lamina. So that makes this what? What organ is this? Muscular artery. And this layer would be what? Tunica media. What type of tissue do we find there? Smooth muscle. Okay, well we've got another section right here, and right out here, what is that layer? External elastic lamina or membrane. Okay, let's see. Let's go on to the next one. We have all three layers right there, and note that that tunica intima in these muscular arteries is going to be thinner than what you see in an elastic artery. Okay, how about this tissue we see right there? Locular fat. Okay, and this is another muscular artery. And so is that one right there, but that's getting pretty close to being something else. But it looks like we've got 1, 2, 3, perhaps 4 layers of smooth muscle. So if we go on to the next slide, what is this one? That's also a muscular artery. We still have an internal elastic lamina. We have multiple layers of smooth muscle in that tunica media. What is this structure right there? It's a nerve and cross-section. Okay, this organ right here, structure, that's an arteriole. That would make this a what? Venule. And let's see, let's go on to the next slide. How about this right here? Capillary. And that's the nucleus of what? A pericyte. Okay, this tissue. What is this tissue? Cardiac muscle. What's What's that structure right there? Intercalated disc. And how about this tissue? What is this? Cardiac muscle and cross-section. See the intercalated disc there, and those nuclei are centrally located, as you can see right there. And it still has that kind of pebbly looking appearance and cross-section, as does skeletal muscle. And that layer that we see right around that is what? Endomysium. Okay, how about these cells? Okay, we've got cardiac muscle right there, so these cells are what? Purkinje fibers. They're cells, but since they're in muscle, they are called fibers. Okay, this tissue, what is this? Cardiac muscle again, right. Okay, and here are some more Purkinje fibers right here. In fact, this is the one that was up on demonstration. Okay, and how about this tissue? What is this? Cardiac muscle again. How about this tissue right here? Smooth muscle. Okay, very good. And this tissue? Skeletal muscle. Okay, now this is a little bit unfair since this is taken with Tomarski interference microscopy. What is that dark band that you see right there? A band. And the light? I band. Okay, and this cell right there, what is that? Okay, that would be a multipolar neuron. And the little specks that we see there, what are those? Missile substance. And what is that thing right there? Right, nucleolus. There's the nucleus right there. Okay, and this is what? This entire thing. Okay, that'd be a peripheral nerve and cross-section. And this layer right there, who said perimysium? Perineurium. And that layer out there would be what? Epineurium. And let's see if we have a little bit higher magnification here. Yes. Okay, so each one of these would be a myelinated fiber. Okay, and so that nucleus right there would be the nucleus of what cell? Schwann cell. How about that cell? What is that? Okay, that is a pseudo-unipolar neuron. We can't really see the stalk coming off of that, the one process. So we would probably accept neuron for that too, but hedge your bet and call it a pseudo-unipolar neuron. And the cells we find surrounding that are what? Satellite cells. So this is from what sort of organ or structure? Okay. Ganglion. Spinal ganglion. And how about this structure we see right there? Node of Ranvier. Or as they say out in Alliance, Ranvier. Okay. And how about that cell right there? Pyramidal cell. So that makes this what? Cerebrum. Okay, that's the only cell we're going to have to identify in the cerebrum, but you need to identify this as an organ. And another slide of the same thing. There's a nice pyramidal cell right there. Large pyramidal cells.
We'll just call it bed cells.
Bed cells?
Bed cells?
Yeah, that would be okay.
We need to differentiate between the layers or not?
No. No.
Are they always called bed cells, or is that just depending on the layer?
No, there is a layer that has the large pyramidal cells. Those are bed cells. Same thing. Large pyramidal cell, bed cell. Okay, what is this organ? Cerebellum. And that layer would be what? Molecular layer, and this would make that the, and this is what? White matter. Okay, and then right here we have a what? Purkinje cells. Now, Purkinje cells could be put on the lab practical even with either a H&E or perhaps with some sort of metallic staining. And those ones that have stained with metallic staining are going to show more in the way of the processes. So these are several Purkinje cells right here, sending their processes up into that molecular layer. And with that gold toning stain, this is what you're going to see. So there is the Purkinje cell, and there's that huge dendritic tree that goes up into the molecular layer. Very pretty slides. Okay, and this organ. And right down here we have what? Okay, that is the dorsal horn, and right in the middle we would find the central canal with the ependymal cells, but we don't see that in this particular slide. Now, if this were a course in neuroanatomy, you would be responsible for all the different clusters of cells and all the different tracts that you see around the outside here. And how about this tissue right here? Simple squamous epithelium. If you said just simple squamous, that would be wrong. And how about this tissue right here? Simple cuboidal epithelium. And this tissue. And that layer that we see right there, what's composing that layer right there? Cilia. And 9+2, yeah. And then we have the basement membrane down here, and later on we'll be looking at the different layers in the trachea. This was taken from the trachea, and we've got a little bit of debris that's being transported by that ciliary escalator. Okay, and there's that basement membrane right there. Has some shrinkage in this particular slide that kind of accentuates that. That's that pseudostratified polymer epithelium. There's that ciliated border. And how about this tissue? Transitional epithelium. Let's go back a couple slides. What's this cell we see right there? Goblet cell. Okay, and more cilia up there. Let's go on to the next slide. And how about this tissue? Simple columnar epithelium. Now we do see some other nuclei here. Those are lymphocytes that are migrating through. So this is simple columnar epithelium. These are not really part of the epithelium. This is just transient. Okay, and right here we have a cell. What is that? Goblet cell. And this tissue, again, is what? Simple columnar epithelium. Okay, what's this tissue? Skeletal muscle. This is one of those teased fibers. This is an entire fiber that's been laid down on a microscope. And this tissue, skeletal muscle in section. You see the striations, you can see the connective tissue investments, and those peripheral nuclei. And there again we see the I band, and there's the, I'm sorry, that's the H band. There's the A band right there. There's the I band, and that's the Z line. This is from an insect, so these are kind of accentuated. You won't see them nearly that clearly in the laboratory. Okay, this tissue right there, what is that? That's skeletal muscle and cross-section. And this layer right around there is what? Perimysium, and around each fiber we have endomysium. Okay, and what is that structure right there? Motor end plate, yeah. Motor end plate. How about these things? What is this? Reticular fibers. And this tissue right here, what is that? Stratified columnar epithelium. And we've got cell right there and one right there. What are those cells? Plasma cells. What do they secrete? Antibodies. And what is this tissue right here? Okay, well that's one you can look up tonight. What is this tissue? Stratified squamous epithelium, non-keratin. And this tissue, unilocular fat, unilocular adipose tissue, white fat. Yeah, all those answers would work. And how about this tissue right here, same as this one and this one? Dense regular connective tissue. Those nuclei belong to what cell? Fibroblasts. And how about that cell? Mast cell. There's another one. And what may be contained in those granules? Yeah, all of those. And this tissue is what? Dense irregular connective tissue. Okay, and this tissue right here? Loose connective tissue, and that would be dense irregular connective tissue. And the same thing, this is overstained with H&E. There's dense irregular, and here is loose connective tissue. What are those fibers? Collagen. Collagen type 1 to be specific. And this tissue, what is this? Hyaline cartilage. And out here we have a layer, that layer would be what? Perichondrium. And this cell that we see right there, what is that? Chondrocyte. And the chamber in which that is residing is called a lacuna, right? And this would be what? Exogenous group. And when you see kind of a difference in staining, that would be what? Territorial matrix. And the only reason it's called that is because it stains a little differently.
Is it the lighter?
Darker.
The darker.
Yeah. How about this tissue? Elastic cartilage. And out here we have what? Perichondrium. And how about this tissue? Fibrocartilage. And what is this cell right there? Right there. Osteoblasts. How about that cell? Osteocyte. What is this lighter layer that we see right there? Osteoid. And this tissue would be what? Bone. Specifically, that would be spongy bone or trabecular bone. And this cell right there, what is that? Yeah, another one right there. So osteoclast. And how about this tissue right here? What is this? Skeletal muscle. And right here, what is this tissue we have here? A little unfair. You can't really see the osteons very well, but this would be lamellar bone. And this layer right here, what is that? Periosteum. In fact, we've got a line of cells right there that are what? Osteoblasts. Okay, they were progenitor cells, except they've now assumed the profile of a osteoblast. So this is all periosteum, and just inside the periosteum we have what? Outer circumferential lamellar. I will guarantee you that somebody's going to mix those two up on a lab fracture. Okay, and what is this thing right there? What is that? Osteon. And right in the middle is what? And these are what? What kind of cells? Osteocytes. And a thin layer right there is what? Endosteum. Okay, and then we have red bone marrow down there. In fact, there is a megakaryocyte, looks like right there. How do I know?
Yeah.
I've been doing this for about 40 years. Okay, how about that right there? Volkmann's canal. Yeah, Volkmann's canal going from reversion canal to reversion canal. There's a little short portion of one right there. And how about this thing you see right there? What is that? Cementing line. So there's an osteon. There's an osteon. What is this right in here? Interstitial lamellae. Okay, what kind of section is this? Round bone section. And that right there is what? Conversion canal. What are all these little fine lines? Canaliculi. And these dark spots are what? Yeah, these are all lacunae. One lacuna, several lacunae. And you can see the lamellae going around that circumference of that osteon. So there's an osteon. There's an osteon down there. There's another one over here. So right in here we have more interstitial lamellae. What's another name for osteon? Conversion system. Okay, what process is going on in this slide? Right, endochondral bone formation. So if we divide this up into zones, this zone is what? Zone of resting or reserve cartilage, and then we have zone of proliferation, and then down here, this is what? Zone of hypertrophy or zone of maturation. And then right in here, we have what? Zone of calcification or the zone of calcified cartilage. And then down here, we have zone of ossification. Okay, so the zone of hypertrophy, zone of calcified cartilage, and the calcified cartilage is going to look a little bit dark. And then as we come down here, we see some new bone that's been laid down in there. And so this would be the zone of ossification. These do not necessarily follow a nice, regular boundary. They can go up and down. What is this organ? Lymph node. Good. And so out here we have what portion of the lymph node?
Cortex.
Cortex. And down in here we have the medulla. What are these things out here? The entire thing would be what? Lymphatic nodule, and the pale center area would be a terminal center. And out here, what is that line that we see? Okay, that's the capsule. Okay, so we've got the nodule right here, so this is all cortex. There's the capsule. How about this vessel that we see out here coming through that capsule? Okay, that would be an afferent lymphatic. And then we can see a portion of the capsule coming down that forms a what? Tuberculum. Okay, let me get the other carousel up. Okay, what is this right here?
Cortex.
Cortex, and that is the medulla. And then we have the capsule. And how about this organ? Have it up here, have it right there. What is this organ? Palatine tonsil. Okay, you see the stratified squamous epithelium. That structure right there is what? Crypt. Okay, there's a crypt. Okay, and so if we look right there, we can see the capsule. So this area is what? Subcapsular sinus. And then coming down along the trabecula would be what? Trabecular sinus, or cortical sinus. And down here, what would those be? Medullary sinuses, and the areas in between the medullary sinuses are called what? Medullary cords. You see that nice capsular sinus right there, and then the trabecular sinus. So the flow is going to percolate through all this material along there, finally getting down to the medullary sinuses. And how about that tissue right there and right there? White fat. Oftentimes there'll be fat associated with these lymph nodes. Okay, and the same thing right there. Nice trabeculum coming down. And in actual fact, when you look at these paraffin sections, there is some shrinkage that actually accentuates those and makes those easier to find. But they are there. And this organ. Well, what's that tissue right there? Stratified squamous epithelium. And we've got what right here? Lymphatic nodule, so that makes this what? Palatine tonsil. And if we look at this epithelium up here, we can see that that is not a stratified squamous epithelium, but it is a pseudostratified columnar, so that makes this organ what? Pharyngeal tonsil. There's the nodule and the germinal center right there. How about this organ? What is this? Thymus. And this area would be what? Medulla, and that makes this the cortex. Okay, and let's see what I wanted to show on that one. What is this structure right there? Hassel's corpuscle. This, I would not show it at this high magnification. This previous slide was supposed to show Hassel's corpuscle, but it's kind of washed out. So there's Hassel's corpuscle. How about this organ? Now, if this were on the lab practical, and it said identify organ, you may move slide, you may change magnification. First thing you want to do is go down to a lower magnification and look around. The person in front of you would probably use higher magnification and then just lift the microscope. So go back down to a lower magnification, scan around the slide, see what's there, then use progressive magnification to make your differentiation. So this organ is what? Spleen. And so right here, what subdivision of the spleen do we have? White pulp, and that makes this stuff out there red pulp. Okay, and so we have that out there. What is that? Capsule. We've got a trabeculum coming down, more or less like fingers. There's white pulp right there, and if we take a closer look at that, what is this entire structure that we see right there? Splenic nodule, and this would be a germinal center, and right there, what is that? Central artery, and surrounding that we have periarterial lymphatic sheath. On a lab practical, I would accept P period, A period, L period. Okay, so this is, collectively, this would be white pulp, and that would make this out here red pulp. And what is that structure we see right there? Splenic sinus, okay, or venous sinus. And between the venous sinuses, we've got splenic cords, or Billroth's cords. Okay, so spleen again, a little bit better differentiation of the stain. There we have white pulp, we have red pulp, and in this case, they probably closed off the splenic vein to back up some blood in these venous sinuses, so they stood out a little bit better. We don't have slides like that that are quite as good as showing that in the laboratory. And again, we've got another central artery right there. We've got a splenic nodule, nice germinal center. And if we go on to the next slide here, this is a plastic section, and there's one of those venous sinuses right there. And it's got those stave-like endothelial cells. And if we go on to the next slide, I think that's probably the last on the lymphatic system. What cell do we have right there? Neutrophil. And how about that cell right there? And that cell. Yeah, band neutrophil. And what are these things? Platelets. Now if you look at this slide, the red blood cells that we see here, oh, what is that cell? Red blood cells. See these red blood cells? Note the color. The eosinophil is going to kind of mimic that a little bit darker, but these are still kind of dingy as compared to a really nice blood smear stain. So we've got kind of dingy red blood cells. So that'll make the eosinophil granules a little bit dingy as well. That cell, that is probably a monocyte. I would not put anything that would be argumentative on the lab practical. Okay, this cell would be what? Another neutrophil. And how about this cell? What is that? That's an eosinophil. Okay, we have a bilobe nucleus. Look how dingy these red blood cells are. So they're going to mimic that. Okay, well, how about that cell? That's an eosinophil too. Okay, how about that cell, smart guys? Basophil. And that cell right next to it, what is that? Lymphocyte. Okay, and that cell, what is that? Lymphocyte. You can see just a thin rim of cytoplasm going around that little indent in the nucleus, ice blue, clear cytoplasm. That is a small lymphocyte. And how about this one? Look at the staining of the nucleus right there. That is a lymphocyte as well, a little bit larger. And how about these cells? Those are monocytes. Textbook monocytes, horseshoe-shaped nucleus, a much finer texture to the nucleoplasm, kind of a foggy cytoplasm. Those are textbook monocytes. And how about this cell? Megakaryocyte. Megakaryocytes that we'll be looking at would be in section. This is a smear. Okay, I think, yeah, that was the last slide.
Do you have an example of a basophilic erythroblast?
I'm sorry, what?
A basophilic erythroblast?
I'm not going to put any of the blood series on the live graph. Any questions? Yes.
If you're asking for like a germinal central nodule itself, how would you differentiate what you're looking for?
Okay, if I wanted a germinal center, I'd put the pointer right in the middle of the germinal center. I'd say identify the structure, be specific. Your answer, just to make sure you get everything right, or get germinal center right, you could say lymphatic nodule germinal center, something like that. You know, pointer on germinal center. So if you're confused, you know, be as specific as you can. Now during the lab practical, you come up to a station and that pointer is pointing to nothing. or you're not sure if it's on the right structure or not, raise your hand. We'll stop the clock. I'll come over and look at it and put it back where it should be. Now, they will get bumped from time to time. So I'll put the number up on the blackboard, which numbers have been moved, so make sure you go back and take a look at those a second time. If you go back a second time and it's not in the same place, let me know right away so we can make sure it's in the right spot. Now, when you're done and you turn in your test, leave. Okay? Don't hang around the door. Leave. Go away. Okay? Go down and get a candy bar that'll be in the oral biology office, or I'll try and maybe I can put them outside the door. Yeah.
How long are you trying at each station?
1 minute for each station, which is plenty of time if you know the answer. I'm sorry, what? Just a few. Okay. I'll have enough extra questions so we get it up to 50 points. Yeah. Question? Okay, if I say identify cell, then it's the osteocyte. If I say identify structure, then you say lacuna, or osteocyte in lacuna, or something like that. Okay. Now, be careful when you write your specific question. If I put the pointer on simple cuboidal epithelium and you're trying to cover all your bases, and you put simple cuboidal or simple columnar epithelium or something, it's got to be one or the other. So, yeah.
Do you get extra time at the end?
Yeah.
You do?
Yeah. I'll probably set the clock for another 25 minutes. If people seem to be struggling, I may set it for a little bit longer. So, how long does Dr. Harn give you?
An hour.
I'm talking about the live graduate.
30 minutes.
30 minutes? Yeah. No, the written test, I'm not sure what the record is. It's something like 12 hours or something like that. So that was written test. So. Yeah.
I'm assuming with like the slides where we can move it will be marked.
Yeah. Yeah. So when you come up on the little post-it note, we'll have the number. We'll say identify cell, identify tissue, identify structure. What does this cell secrete, or something along those lines. And then it'll say do not move. Okay. That doesn't mean you. It means don't move the microscope or anything else. If it's an organ identification, like if I put the spleen up or something, and I say identify organ, I'll say you may move, slide, and change magnification. Now, when you're up there, don't move the eyepieces. You can change the distance between eyepieces, but don't twist the eyepiece because they will be fixed in place. Yeah. Yeah. Fine focus. You can move fine focus. That's probably all you'll need to use.
For all of
Yeah. Yeah.
And this is probably stupid, but you want us to stay at our station until the time goes off and then, right?
Right.
Okay.
Right. Now, after we've gone through once, and you guys are going around to check stations a second, third, fourth, fifth time, okay, you can go there for 1 minute, okay, and then let the person behind you have a shot at it. You can come back, but don't hog the microscope. I mean, after you look at it for a minute, the answer is not going to come, that much quicker. Okay, so look at it for 1 minute. Let the person behind you have a shot at it. Go look at another microscope. You can still come back and look at it, but only 1 minute at a time. Okay, any other questions? Okay, this will be fun. Okay. The only thing that I have not been able to do on these lab practicals, and you guys can probably help me for this for next year's class, is I still can't figure out how to put a black box question like Dr. Harn puts in his lab practical. Okay, now we'll aim to start at 1 o'clock. If we haven't got it completely set up, have a little patience, because it's just Phyllis and me setting that up tomorrow. And, yeah. If you guys want to help, after everybody's done, you can help us put the microscopes away. Okay, we'll see you tomorrow at 1 o'clock.